Hello guys, welcome to our channel AskPrep. Today's series is for 11th class biology. We will be studying chapter 9, Biomolecules in detail. We have also added important notes for your exam preparation. All the best for your exam. Starting with introduction. What is biomolecules? Biomolecules are essential organic compounds produced by living organisms. They primarily consist of carbon, nitrogen, hydrogen, phosphorus, oxygen, and sulfur. The four major types of biomolecules include proteins, carbohydrates, nucleic acids, and lipids. These molecules play vital roles in biological processes, contributing to life functions and organism structure. Analysis of chemical composition. Organic compound analysis, proteins, nucleic acids, carbohydrates, uses trichloroacetic acid. The tissues are ground to form a slurry for better analysis. Inorganic compound analysis, minerals, metals, involves burning tissues to form ash, leaving only the inorganic components. Next is proteins. Proteins are nitrogenous organic compounds made up of amino acid chains. Amino acids are composed of hydrogen, carbon, oxygen, and nitrogen, with 22 types found in nature. In water, amino acids exist as dipolar ions, zwitterions, capable of donating or accepting protons. Amino acids exhibit optical activity due to their chiral carbon, except for glycine. This is an introduction protein. Let's understand in detail.
Protein is an essential part of human diet. It's found in a variety of foods like eggs, dairy, seafood, legumes, meats, nuts, and seeds. Regardless of the source, the protein that we eat gets broken down and reformed into new proteins in our bodies. These proteins do everything from fighting infections to helping cells divide. You name it, they are doing it. At its simplest, a protein is a chain of amino acids bound to one another by peptide bonds. Like a string of beads, these strings get twisted and folded into a final protein shape. When we eat protein, it gets broken down into its individual amino acids. Most amino acids have a central carbon atom bonded to one amino or nitrogen containing group and one carboxylic acid group. That's why it is called an amino acid. The carbon also has one hydrogen atom and a side chain which is unique to each amino acid. The exception to is this proline, which is a tiny little wing structure. Instead, although there are hundreds of amino acids in nature, humans only use about 20 of them to make basically every type of protein. They include alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutenine, glycerin, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, trizine, and valine. That's 20. One way to divide them is by defining which ones our body can make and which ones we can't. There are 5 amino acids: alanine, asparagine, aspartic acid, glutamic acid, and serine that we can get from foods, but we can also make ourselves. These are called non-essential amino acids. Then there are 6 that we call conditionally essential because healthy bodies can make them under normal circumstances: arginine, cysteine, glutenine, glycerin, proline, and trizine. But we can't make them in cases like starvation or certain inborn errors of metabolism. Finally, there are 9 of them that we can only get from food: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. We call these the essential amino acid. Dietary protein provides the essential amino acids that are needed to make our own protein, hormones, and other important molecules, a circle of life of sorts. But to do so, we need to break the dietary protein down first through a process called proteolysis. When we first eat a protein containing food, proteolysis begins. When food reaches the stomach, first hydrochloric acid denatures the protein, unfolding and making the amino acid chain more accessible to enzymatic action. Then pepsin, which is a protein itself made by gastric chief cells, enters the picture. Pepsin cleaves any available protein in small oligopeptide chains which move into the duodenum, where a second set of digestive enzymes made by the pancreas further chop the oligopeptides into tripeptides and dipeptides and individual amino acids. These can all be taken up to the essential cells where di and tripeptides are then converted into amino acids. Some amino acids remain in these cells and are used to synthesize intestinal enzymes and new cells, but most enter the bloodstream and are transported to other parts of body. In general, animal-based protein foods like egg, dairy, seafoods, and meat provide all 9 essential amino acids in adequate amounts. Soy foods are unique in that they are plant-based and also provide all 9 essential amino acids in adequate amounts. Most other plant foods, including whole grains, legumes, nuts, seeds, have high amounts of some amino acids and low amounts of other. Hearing this, it might be easy to assume the animal-based food provide more protein than plant-based ones. But as it turns out, a cup of tofu and has the same number of grams of protein as 3 ounces of steak, chicken or fish, and a half a cup of lentils has more grams of protein than an egg. And not all plant foods are low in the same amino acids, so eating a variety of plant-based food can provide all 9 of the essential. For example, pairing protein source like rice and beans, or a hummus and pita bread, or oatmeal topped with almond butter. However, in terms of volume, it may be necessary to eat more plant-based foods to get a similar amount of protein and amino acid profile provided by animal-based proteins. Generally speaking, daily protein requirements are based on studies that estimate the minimum amount of protein needed to avoid a progressive nitrogen loss. The World Health Organization guidelines and the US recommended dietary allowance each estimate that daily protein requirements for healthy adults are about 0.8 grams per kilogram of body weight. Protein recommendations per day vary by age as well. Children 1 to 3 years of age are recommended to get 13 grams. For age 4 to 8, 19 grams are recommended, and between ages 9 and 13, 34 grams are recommended. Whether a person is male or female also impact protein needs. Female ages 14 and above are recommended to 46 grams of protein per day. Males age 14 to 18 needed slightly more, about 52 grams per day. In males 19 and older are recommended to get 56 grams per day. Some groups like pregnant and breastfeeding women as well as athletes have elevated needs, and older adults may also benefit from eating more protein. Getting the right amount of protein per day can be achieved in multiple ways. For example, 1 serving of Greek yogurt with breakfast, a salad topped with 3 ounces of chicken for lunch, and 3 to 4 ounces of a fish at dinner provides about 64 grams of protein. Getting the same amount of protein could also be achieved by eating a cup of tofu scramble and a slice of peanut butter toast for breakfast, a cup of shelled atame with lunch, and 1 cup of lentils and brown rice at dinner, which also offers about 64 grams of protein in total. Now the fact is that it is still unclear what an optimal amount of protein is, and the research is ongoing in clinical settings. These are certain individuals who may be at risk for protein deficiency, including patients with malnutrition, trauma and burn injuries, as well as various conditions impacting nutrient absorption like inflammatory bowel disease. These individuals may have increased protein needs compared to the general population. Except for certain circumstances like kidney disease, isn't a health risk associated with eating a lot of protein because our bodies are able to process it? Alright, as a quick recap, protein is a cornerstone of the human diet and a major component of our bodies. There are 5 non-essential, 6 conditionally essential, and 9 essential amino acids. We need to get essential amino acids from our diet. Protein needs vary depending on a life cycle stage, level of physical activity, and health status. Some health conditions may put people at risk for protein deficiency or increased protein needs. Everyone, whether omnivorous, vegetarian, or vegan, can get enough protein by eating a variety of food.
Next up is structure of protein. Peptide bonds link amino acids together in proteins. Levels of protein. Organization. Primary structure, the linear sequence of amino acids linked by peptide bonds. Secondary structure. Protein folding into alpha helices or beta sheets stabilized by hydrogen bonds. Tertiary structure. The three-dimensional arrangement of the entire protein involving hydrophobic interactions, hydrogen bonds, and electrostatic forces. Quaternary structure. Some proteins consist of multiple polypeptide chains forming a functional complex. Example, hemoglobin. Now, let's understand structure of protein in brief.
Let's talk about structure of protein. So the proteins have a primary, secondary, tertiary and a quaternary structure. Let's talk about each of them. Primary structure of protein. So the primary structure of protein is a linear sequence of amino acids present in the protein. This linear sequence of amino acid is either written in a 3-letter or a 1-letter form. Now these amino acids are attached with each other by a peptide bond. The primary structure of protein is always written from the N-terminal of the first amino acid to the C-terminal of the last amino acids. Information about the primary structure of protein can be obtained by sequencing the gene that encodes that protein. This information can also be obtained by sequencing the protein itself. Secondary structure of protein. Now, in the primary structure of protein, the amino acids are linked by the peptide bond, and the peptide bond has an oxygen attached with the carbon and a nitrogen attached with the hydrogen. Both oxygen and hydrogen have the ability to form hydrogen bond, and this results in folding of the peptide chains either in alpha helix or the beta sheet. Now let's talk about the alpha helix. Alpha helix is the most common secondary structure of protein, and the structure the protein attains a spiral shape such that the side chain of amino acid remains outward. Helix is stabilized by hydrogen bonding. This hydrogen bonding is formed between the hydrogen of nitrogen and the oxygen attached with the carbon. Individual hydrogen bonding are of course weak, but collectively they are strong enough to stabilize the spiral structure. While the peptide bond except the first and the last participate in the hydrogen bonding. Each turn of the helix has 3.6 amino acid at a distance of 0.54 nanometers. Presence of amino acids like protein can make the helix unstable and disrupt the helix. The beta sheet. Beta sheet is the second type of secondary structure of protein. Now in alpha helix, the hydrogen bonding was between the same peptide chain, while in the beta sheet, the hydrogen bond is formed between the two different polypeptide or the same polypeptide chain. The beta sheet can be either arranged in parallel or anti-parallel fashion. Structure of protein. Arrangement of protein in three-dimensional space is the tertiary structure of protein. The torsion structure will also have other bonds like disulfide bond, hydrophobic interaction and ionic interaction. Quaternary structure of proteins. The proteins having tertiary structure can further interact with other tertiary structure proteins to either form dimers, trimers, tetramers and so on. The structure so formed is called quaternary structure. Most of the protein in the cell have the quaternary structure.
Structure of amino acids. Amino acids form peptides. 2 amino acids create a dipeptide, 3 form a tripeptide, and 12 to 20 make oligopeptides. multiple amino acids link to form polypeptides. The first amino acid is the N-terminal, amino terminal, and the last one is the C-terminal, carboxyl terminal. Next is nucleic acids. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are essential biomolecules. Nucleic acids are composed of nucleotides, which consist of a nitrogenous base, a 5-carbon sugar, and a phosphate group. Nucleotide structure. Nitrogenous bases are aromatic molecules with heterocyclic structures. Purines, adenine, guanine, and pyrimidines, cytosine, thymine in DNA, uracil in RNA are key components. Next is DNA or deoxyribonucleic acid. DNA contains deoxyribose, a 5-carbon sugar, while RNA or ribonucleic acid contains ribose. When a nucleoside, which consists of a nitrogenous base and a sugar, is combined with a phosphate group, it forms a nucleotide. DNA structure. bDNA. Antiparallel strands with adenine thymine and guanine cytosine pairs held by hydrogen bonds. zDNA. A thinner structure alternating purine pyrimidine bases stabilized by high salt concentration. You can see the image. Moving with RNA or ribonucleic acid, it has 3 types of RNA. 1. mRNA or messenger RNA carries genetic information for protein synthesis. 2. tRNA or transfer RNA transfers amino acids during protein synthesis. 3. rRNA ribosomal RNA forms ribosomes for protein synthesis. Next is carbohydrates, composed of carbon, hydrogen, and oxygen. Carbohydrates include polyhydroxyaldehydes or ketones. Monosaccharides, simplest carbohydrates, example glucose. Disaccharides, formed by 2 monosaccharides. Oligosaccharides, polymer of 2 to 10 monosaccharides. Polysaccharides, polymers of hundreds to thousands of monosaccharides. Example, starch, cellulose, glycogen. Some common saccharides, starch, plants, and glycogen, animals, store energy composed of glucose units. Starch has 2 forms, amylose, unbranched, and amylopectin, branched. Glycogen, mainly in muscles and liver, is highly branched. Structure of starch, cellulose, structural component in plant cells, unbranched glucose polymer. Chitin, found in insect crustacean exoskeletons, made of N-acetyl-D-glucosamine residues. Monosaccharides can reduce ferric or cupric ions, while non-reducing sugars, example sucrose, cannot. Lipids. Lipids are water-insoluble but soluble in non-polar solvents like chloroform and ether. Functions of lipids: energy storage, stored in adipose tissue as a long-term energy reserve. Membrane structure: form the lipid bilayer in cell membranes, regulating molecule passage. Protection: lipids in skin reduce water loss and protect against pathogens, and plant waxes provide a protective coating. Next is fatty acids. Fatty acids are long hydrocarbon chains with a carboxyl group at one end, either saturated or unsaturated. Unsaturated fatty acids, example linoleate, are essential and must be obtained from the diet. Triacylglycerols, triglycerides, non-polar molecules made of fatty acid esters and glycerol, essential for energy storage. What are enzymes? Enzymes are proteins that catalyze biochemical reactions. They exhibit primary, secondary, and tertiary structures with an active site that binds substrates. Enzymes are specific to substrates, acting as catalysts without being consumed in reactions. Types of enzymes: oxidoreductases, transferases, hydrolases, lyases, ligases, and isomerases. Cofactors and coenzymes. Cofactors: non-protein molecules activating enzymes. Coenzymes: organic compounds that bind transiently. Prosthetic groups: tightly bound organic substances within enzymes. Factors influencing enzyme activity: temperature and pH. Enzymes have optimal conditions for activity. Inhibition. Competitive inhibitors, example malonate, compete with substrates for active site binding. Thanks for watching the video. We will drop the notes in the description of this video. If you found this video helpful, please like, subscribe and share. See you in the next video and all the best for your exam.